So I'd like to talk about something we refer to as the resting potential. And the resting potential is the state of the neuron when it is not firing any kind of electrical message. There's no kind of long-range electrical message, no sodium current, heading from one end of the neuron to the other when the neuron is at what we call rest. And resting means there's no current flowing. Potential is a funny term. Potential actually is part of a larger term. It's potential difference is what it stands for. And what it means is there's a difference in charge, in the sort of relative charge inside the neuron versus the out. So that membrane, remember, is impermeable to the passage of charged particles. So it's selectively permeable to some and doesn't let others go through, partly, of course, dependent on which protein machines are located in that membrane that can move things back and forth, right? So at rest, there's a particular charge difference across that membrane that is maintained with some ions out here and some ions on the inside. Ultimately, we want to have sodium ions, head down the neuron to carry the electrical charge, the electrical current, when the neuron fires an information-rich action potential. We want to store sodium to a certain extent. So we're going to see that sodium is actually found in great concentration outside the membrane, which should suggest to you, that at rest, the membrane is not very permeable, to the passage of sodium, and that's going to be true. So let's look closely at this this representation of a neuron's membrane. Now, you have to look at your overhead here, but I'm gonna hopefully you should be able to identify the following constituents here. You have the little ball and stick kind of molecules that form, of course, our phospholipid bilayer. Right? This is the impermeable, kind of membrane that surrounds every cell, including neurons. And then embedded in that phospholipid bilayer are a series of protein machines, right? And there are a couple of protein machines that you should see illustrated there that are showing ions moving through them. They're literally just holes in the membrane. And these are channels. They're specific protein channels. They are protein channels that are selective, which means they only let certain ions go through and not others. They're sort of got a very specific kind of passageway or pore that is shaped to allow only some ions to pass through, but not others. So you should see two potassium ion channels that are illustrated here, and one sodium ion channel that's illustrated here. And what you should look at and note is that the potassium seems to be leaving, going from the inside of the cell and passing out, while the sodium appears to be coming from the outside and heading in. These are what we refer to as ungated channels, the sodium and potassium channels that are indicated here. They're open all the time. It doesn't matter, if the membrane potential changes or anything else changes, these are open all the time. And because they're just holes that let these specific ions go through, the direction of the ion flow is the way the ion wants to go. So forces are aligned to drive it, and the potassium is going out because it wants to go out. The forces are pushing it out, and sodium is coming in because there are forces that are sort of set up to pull it in, right? So it's not like these channels are transporters. No energy is required to get the ions to move in the directions that they're moving because they're naturally driven by forces. Now, what are the forces? Well, there are three forces that act on ions, and I want you to know these very well. One is the electrical force. This is actually listed as number 2, but we talked about the electrical force. Oppositely charged, ions attract each other, right? Well, like charges repel. So that's one reason why an ion will move from one location to another, because it's either repelled by like charges or it's attracted by some opposite charges. Another force, number 1 here, is the concentration gradient force, and a gradient is just a change from a lot of something to a little of something. So a concentration gradient is you've got a lot of something, let's say an ion, let's say a lot of sodium over here, and then you got very little sodium over here. Well, the concentration gradient force states that something in high concentration will tend to move to areas of lower concentration, and it's a natural kind of phenomenon in that anything with a temperature is moving, right? And if it's in high concentration over here, these are all little random motions, it'll tend to distribute into areas of lower concentration. It would be very strange to have something in high concentration, so just keep making movements that kept it in that same location. It would tend instead to sort of randomly distribute into areas of lower concentration until ultimately everything's sort of at equilibrium, right? And the final force is this final machine, this protein that is embedded in the membrane that's shown on this image here in your overhead. And this is it's referred to as a pump, but I want you to think about what a pump really is in terms of the terms that I've given you for protein machines. A pump this is called the sodium-potassium pump. This particular biological machine that you generate, based upon genetic instructions that you transcribe and then translate into, this very complex protein that sits in your membrane, grabs onto 3 sodium ions from the inside of the cell, okay? And then it grabs on simultaneously to 2 potassium ions from the outside of the cell, and then ATP comes along, right? Their little chemical battery, and the bond is broken and the whole pump shifts. It shoves the 3 sodium ions out of the cell into the extracellular space, and it pulls the 2 potassium ions inside the cell. And the terrific thing about this particular machine and this particular force really is that it establishes 2 gradients. Okay? Now think about this. It first of all establishes a concentration gradient. It puts more sodium on the outside of the cell, right? And there's an open channel that lets sodium come through, right? But there aren't too many of those. The cell membrane at rest is not very permeable to sodium. So it establishes a very high concentration of sodium on the outside, and that's indicated by the Na+ that you see in the extracellular space with big brackets around it. That means a lot of concentration of sodium. And then on the inside, there's a very small concentration of sodium because that pump, it turns out, is quite efficient and kicks a lot of sodium out. So sodium ends up being sort of masked on the outside of the cell, and it can't really get back in because that membrane is not very permeable to its passage. It also establishes a pretty significant potassium concentration gradient, right? Because it's bringing potassium inside the cell. And potassium that comes inside the cell can leave. There are some there are more of these open channels that let potassium flow, and it will flow, but still the pump is pretty efficient, so you end up with a much higher concentration of potassium on the inside of the cell than you have on the outside of the cell. Now, so the first gradients that it establishes are these concentration gradients, but the second gradient it establishes, I want you to really think about this, is an electrical gradient. We have, 3 positively charged sodium ions going out for every 2 positively charged potassium ions coming in. So you're kicking out 3 positive charges for every 2 that you bring in. So think about where you're getting an accumulation of more positive charges. You're going to get more positive charges, relative to the inside there, set up on the outside of that cell. So this is establishing also a charge gradient or an electrical gradient across the membrane. So in a membrane at rest, you have more potassium inside because of that sodium-potassium pump. You have more sodium outside because of the pump and because of the lack of permeability of that membrane. And you have more anions. Remember those proteins, right, that tend to have a negative charge on the inside as well. So if you compare the charge across the membrane, the inside is about 65 millivolts. That's 1,000. Those are thousandths of a volt, or 70 millivolts, depending on the species and the location. More negative inside than it is outside.